The IMViC tests are a group of 4 tests that are always done together. Their pattern of whether they're positive or negative help identify gram-negative bacteria, particularly those in the family Enterobacteriaceae. The mnemonic stands for I is for indole, M is for methyl red, V is for Voges Proskauer. We throw the little I in there so we can make something pronounceable. And C is citrate. So these are the 4 tests you'll be doing. The indole test checks for the presence of the enzyme tryptophanase. Tryptophan is an amino acid found in protein. If the organism produces tryptophanase, it will break tryptophan down to indole. We can react indole with Kovacs reagent and we get a pink color. The indole test is a spot test. We have a little card that has filter paper squares in it, and the filter paper has been impregnated with Kovacs reagent. You select test colonies with a sterile loop and then smear the test organism on one of the test squares. A pink color is a positive result, and no color change or a yellow color is negative. The methyl red Voges Proskauer is actually 2 tests, but you're going to set it up in one tube. The tube will contain glucose because these 2 tests will check for the type of glucose metabolism the organism can perform. Since this is a broth, you're going to inoculate an MRVP broth, and you want to inoculate it pretty heavily. Then you're going to incubate this for 18 to 24 hours at 37 degrees centigrade. The citrate test is going to check to see if the organism can use citrate as its only carbon source. Citrate is going to be the only carbon source in this media. Since this is a slant, you're simply going to inoculate the slant and then incubate for 18 to 24 hours at 37 degrees centigrade. The MRVP test is really 2 tests. Both determine how glucose is metabolized by the organism. Remember, Pasteur's theory of fermentation was that different organisms will take the same substrate and give you different end products. The substrate in MRVP is glucose. Organisms have 2 possible pathways for glucose metabolism. One pathway is to metabolize glucose to acids and stop. So we say that it's stable acids. They don't change to anything else. Other organisms metabolize glucose to acids and then further metabolize the acids to other end products that are neutral, not acidic. These are things like acetoin and 2,3-butanediol. To do the methyl red portion of the test, we're checking to see if glucose metabolism is glucose to stable acids. Since we can't see this metabolic process, the reaction is invisible, we're going to have to add a reagent to detect the acid end products. To do this test, you'll take 3 drops of the MRVP broth and place it in a well on a spot plate. Methyl red is the reagent. This is a pH indicator. It turns red at an acid pH. You're going to add 1 to 2 drops to the well containing MRVP broth. A red color is positive, a yellow color is negative, and these results are immediate. You'll see this right away. To do the Voges Proskauer test, we're checking to see if the glucose metabolism is glucose to acids and then further break the acids down to neutral end products. Since there won't be acids, we would get a negative MRVP. Once again, we can't see any of these reactions, so we're going to have to add reagents to detect the end products. You'll start again by placing 3 drops of MRVP broth in a well on a spot plate. The reagents for the Voges Proskauer test are 1 naphthol, or sometimes called alpha naphthol, and 40% potassium hydroxide. You will add 2 drops of 1 naphthol to the well with the MRVP broth in it. You will then add 1 drop of 40% KOH to the same well. A yellow is negative, a red is positive, but this reaction takes 20 minutes. So for doing time management, when you're starting your IMViC test, the very first thing you want to set up is the Voges Proskauer. That way you will have time for the reaction to complete while you're doing things like reading the citrate and doing the methyl red test. The citrate test only has citrate in it as a carbon source. If the organism cannot use citrate as a carbon source, it will not grow at all on the medium. Organisms that use citrate release bicarbonate, and this increases the pH in the media to 7.4 or higher. This agar contains a pH indicator, bromethymol blue. At a pH above 7.4, bromethymol blue turns blue, otherwise it's green. So you want to look at the slant. If the slant is blue, you have a positive citrate. If the slant is green or there's been no color change, you have a negative citrate. The IMViC reactions will give you certain patterns. This helps indicate what organism you're dealing with. E. coli always gives a positive, positive, negative, negative pattern. That is, positive indole, positive methyl red, negative Voges-Proskauer, negative citrate. Members of the genus Enterobacter give you just the opposite: negative indole, negative methyl red, positive Voges-Proskauer, positive citrate. Members of the genus Klebsiella give you the same pattern. Wait a minute, you're saying the Klebsiella reactions didn't work out quite that way? Maybe you got a positive methyl red and a negative Voges-Proskauer? Well, you have to remember that that glucose reaction is glucose to acids, and if you test at that point, you would have the low pH and get a positive methyl red. You have to wait for the acids to be converted to the neutral end products to get the positive Voges-Proskauer. Some species of Klebsiella do this reaction very, very slowly, and if you test too early in the reaction pattern, you may get a false positive methyl red. All right, you've got unknowns coming up, and you're going, well, how will I know if I got a false positive methyl red? You're going to have a sheet that will have all of the positive and negative reactions for all of the tests you will have to do. If you go down through there and nothing is matching well, try ignoring the methyl red and Voges-Proskauer reactions and see what you get. Usually, if you've got a problem with the methyl red being a false positive, if you simply take it out of the equation, you still have enough test reactions that you can identify your organism.